Mutations that cause genetic disease can sometimes be diagnosed using enzymes that cleave DNA. In sickle cell anemia, the sickle cell allele has a mutation that converts an adenine base to a thymine base. Whereas the normal sequence is digested by the restriction enzyme MST2, this enzyme cannot recognize the sequence in the sickle cell allele and therefore does not cut it. Both alleles of the beta-globin genes have MST2 recognition sequences in other regions of the gene. If you were to digest both samples of DNA with MST2 and then use a probe to highlight the DNA fragments, how many fragments would you find? Enter the number of fragments that the probe would identify for each allele. Restriction enzymes recognize short sequences, which can usually be found throughout a long stretch of DNA. In the case of MST2, the recognition sequence is 5' CCTNAGG 3', where N can be any base. On average, how often would you expect to find an MST2 recognition sequence along the DNA of a chromosome? Click on the correct answer. In performing this test, you would digest genomic DNA from the test sample of interest with MST2, as well as digest DNA from a normal beta-globin and a sickle cell sample. The digested DNA is then loaded onto an agarose gel for electrophoresis. The DNA fragments are negatively charged and migrate toward the positive electrode in an electric field, with the smaller fragments moving faster than the larger fragments. When genomic DNA is digested, there are usually so many bands of DNA that the individual fragments are difficult to distinguish from each other. To highlight the DNA fragments surrounding the sickle cell mutation, usually the DNA from the agarose gel is blotted onto a filter, hybridized to a radioactive probe, and placed on X-ray film to identify the bands of interest. In this case, the probe is complementary to the fragments surrounding the mutation site. From the information on the X-ray film, what is the genotype of the person from which the test sample was taken? Click on the correct answer.